So in this video, I'm going to take an overview of cellular respiration and break it down into its individual steps. What I might recommend is that you follow along with your own diagram. There's several ways to approach this. One, you can try to diagram the cellular respiration yourself before you watch this video. Use the terms that are listed in the vocabulary part of the week 8 plan. See how well you do on your own, then watch the video and update your diagram as you go. It's actually really good for your learning. Another possibility would be that you listen just to the audio file on this video as you diagram and try to place all of the components that are discussed in your own diagram as you go. And finally, you can just watch the video and diagram as you go. There's part of this physical act of placing the proteins in the specific location in the cells that I think can be helpful to remembering how this process goes. So let me just orient you to this beautiful cell diagram that I just drew. That was sarcastic tone in case you didn't pick up on that. So out here would be my cell membrane or plasma membrane. This is a nucleus. It's not really relevant to the diagram, but I just wanted to make sure you didn't forget that all of these cells have nuclei. And then there should be lots of other organelles, right? There's an endoplasmic reticulum here that we're not really going to worry about. And then I made this mitochondrion here very large because we're going to have to put a lot of stuff in it. And I usually run out of room in these diagrams, but I'll try to set myself up so I don't here. So one mitochondrion, many mitochondria. All cells have many mitochondria in them. The mitochondrion is made up of two layers of membranes. This is the outer membrane. This is the inner. This part inside is called the matrix. This is all an aqueous solution, lots of water and solutes. And then this part in between the two membranes is called the intermembrane space. At least that one makes sense. And then this part out here you're all familiar with already is the cytoplasm. And cellular respiration takes place in all of these different compartments. All right, first step of cellular respiration is typically glycolysis, and that's definitely true if you start out with a molecule of glucose, which is what we're going to do. So glucose would normally come from the outside of the cell. It would have to come into the cell via facilitated diffusion down its concentration gradient. And what keeps driving glucose from the extracellular space into the cytoplasm of the cell is that there's a concentration gradient, because once glucose gets into the cytoplasm, it is acted on by a variety of different enzymes, ultimately which will produce the molecule or two molecules of pyruvate. Right, so glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. So we have to account for the carbons. I'm not going to go into the details of glycolysis, except to say that there are two major steps. The first part of glycolysis actually requires an input of energy. So ATP is used, actually 2 molecules of ATP donate their phosphate groups to glucose, make glucose first 6-phosphate, and then you actually get a bisphosphate molecule, and then it breaks apart. So the two halves of the molecule come apart to make 2 molecules of glyceraldehyde 3-phosphate, which should sound familiar since we saw that molecule in photosynthesis. These molecules can actually pick up some additional phosphate and donate some of their electrons to NAD plus to make NADH, which we'll see later. And then finally, right before the last step, we get some ATP energy back out. So 4 molecules of ADP pick up the 4 phosphates that were added previously to make 4 molecules of ATP, along with the 2 molecules of pyruvate. All of this takes place in the cytoplasm. The next step is actually pretty straightforward, called pyruvate oxidation. And so what it does is exactly as it sounds. Pyruvate enters the mitochondrion at this stage, so it has to move through both the outer and the inner membrane of the mitochondrion. And as it comes into the matrix of the mitochondrion, pyruvate, which again is 3 carbons, will donate some of its electrons to NADH. So we have another NAD plus to NADH here. This is for 1 molecule of pyruvate. Don't worry about the numbers really. And then because this is, at this point, one of the carbon is at its most oxidized stage, you lose a molecule of CO2. So that's 1. We have to account for all of the carbons. So of course, if we have 2 molecules of pyruvate, we would have 2 molecules of CO2 coming out. So that leaves us with a 2-carbon molecule, acetate, which is linked to a coenzyme, which is it's actually a vitamin that we use and we have to ingest. And so we get acetyl CoA. And the acetyl is really the part that matters, and that's 2 carbons. Acetyl CoA is what's added to the citric acid cycle. So the 2 carbons of acetyl CoA get actually added to a 4-carbon molecule called oxaloacetate. Again, you don't have to worry about that name. And transforming it into a 6-carbon molecule called citric acid. So that's where the cycle gets its name. These molecules are further processed through a variety of different enzymes in the citric acid cycle to eventually regenerate the oxaloacetate, so it can take up another acetyl CoA. Note that coenzyme A is regenerated at that point so that it can continue to pick up more acetyl coming in through pyruvate oxidation. Again, we have to account for the carbons, and so as the cycle moves along, carbons are further oxidized, and the last 2 carbons that came in with acetyl CoA are released in the citric acid cycle. So we've accounted for all of our carbons. 2 out of the citric acid cycle, 1 out of pyruvate oxidation accounts for the 3 carbons that came in with pyruvate. 2 rounds of that gives you the 6 CO2 that came in with glucose. Okay, so for every oxidation, there has to be some reduction as well. The electrons that are released by these oxidation steps are picked up by NAD plus, which is reduced to form NADH, and there are 3 molecules of that for every turn of the citric acid cycle. And then just because it's different, there's another electron carrier called FAD that's used in the mitochondrion, and when it is reduced, it becomes FADH2. No matter what, these electron carriers will be carrying their electrons to the next step of cellular respiration. There's a few other products, minor products out of the citric acid cycle. There's some ATP, well, GTP, which is then transformed to ATP. It is such a minor part of the citric acid cycle that we typically don't worry about it. NADH and FADH2 are the primary outputs in terms of moving the energy along in cellular respiration, and then CO2 is the primary matter output of the citric acid cycle. This is typically when I start running out of room on my diagram, and this time seems to be no different. But for the electron transport chain, we now move to the inner membrane of the mitochondrion. All of the prior steps, the last part of pyruvate oxidation and citric acid cycle have been taking place in the matrix. And now we have all of these NADH molecules and FADH2 that are floating around in the matrix. They're waiting to bump into the molecules of the electron transport chain, which reside in the inner membrane of the mitochondria. There are 4 of those big membrane proteins called complexes. They're numbered 1, 2, 3, and 4. Not very creative. They are where electrons are deposited and move through, just like we talked about in photosynthesis. So the electron donors in this electron transport chain are the NADH molecules and FADH2, right? And it doesn't matter where they were generated. NADH can donate to complex 1, and that means it's oxidized to reform NAD+, which can go back and pick up more electrons. So here's an electron moving now down the chain. And FADH2 actually donates to complex 2. Again, not a big deal. So electrons enter here and you regenerate FAD. These electrons will move via proteins through the electron transport chain until they reach the terminal electron acceptor, which is oxygen, right? So oxygen will come in and pick up these electrons at complex 4 and pick up some protons along the way to now be reduced and form water, another product of cellular respiration. As the electrons move through the electron transport chain, as we've discussed before, in other electron transport chain, we'll see movement of protons from the matrix of the mitochondrion to the intermembrane space, such that now there is a higher concentration of protons in the intermembrane space than in the matrix. You should note that by the end of the electron transport chain, we have accounted for all of the matter that came in with cellular respiration. The reactants, glucose and oxygen, have been transformed through a series of redox reactions into CO2 and water. But we haven't accounted for the energy. The energy we've been following along, but right now it's still trapped in this proton gradient across the inner membrane of the mitochondrion. And so this is where the ATP synthase comes in. Oh, I forgot to title my slide. ATP synthase. It doesn't matter where you put it. It just has to be on the inner membrane of the mitochondrion facing the matrix, because now as protons accumulate in the intermembrane space, they will flow through the ATP synthase back into the matrix, allowing the turbine to turn and pick up ADP and phosphate in the matrix, converting it into ATP. And so finally, we have the energy in the form of ATP. Sadly, ATP is used in the cytoplasm. And so at some point, we got to figure out ways to get that ATP back out of the mitochondrion. But I'll let you think about how that might be done.